outer det source. Should do it. Okay, so I realized just before I came to class, I haven't got you guys the figures posted on the PowerPoints for this. I apologize. I will get those posted for you tonight and get you caught up and ahead of where you were. And I see two hands going up as soon as I say that.
We have the same question. When will the video for Monday be posted?
It should be posted later this evening also. Yeah. I usually try to get them up pretty quickly, and I had a big banquet last night, so I didn't get a chance to work on it last night. But tonight, it'll probably be late tonight, but tonight I should have some time to get it posted. Okay? And if not, you can come and cut me or something. I don't know. Was that your question also?
Yeah.
Okay. All right. So we're about done with translation. So I want to say just a couple more things about translation, and then we'll move into some of the applications of what you've been learning. So as I hope you've been sort of thinking about these as we go through, we saw how we took, for example, our knowledge of proteins structure and how we were able to use that knowledge of protein structure in our strategy for purifying proteins. And similarly, we've got other things that we've learned about both proteins and DNA that we apply and use. And the application of those is what we think of as biotechnology. So I want to say a little bit about, in fact, that's most of what I'm going to talk about today. For some reason, the outer projectors are not on, so we'll have to live without them, I guess. So last time I showed you the looping and so forth, I told you that the overall process of translation in eukaryotes and prokaryotes is very similar. So I'm not going to go that there are different names for different proteins and so forth, and it's the process that I'm much more interested in than I am in giving you a bunch of different names to all memorize and do. There are some differences, but from a bigger perspective, the processes are astonishingly similar. Okay? So that's what I will leave you with on eukaryotic translation. Post-translational modifications, we've actually talked about some post-translational modifications already. For example, converting zymogens into the active form is a post-translational modification because it involves proteolytic cleavage. I'd like to show you this one because for anybody interested in human health, it has some important implications. And it's a protein we'll talk about later in the term. It's insulin. And this protein has a very interesting set of modifications that happen to it. It is synthesized first as a single polypeptide chain. And you'll notice it's called precursor of pre-pro insulin. Okay? So we can see that there's a lot of preparation there, all the pre's and all the various things that's happening there. Pro is also another way of saying it's a precursor. So you have a precursor of pre-pro insulin, meaning that this guy's got to be processed a fair amount to make the final mature insulin molecule. Because if it's not processed, it won't function. Okay? So it's synthesized like this, and you can see that what's happening at this point is that the insulin protein is folding, and that folding is bringing together some sulfhydryl groups that make disulfide bonds, as you can see here. And those disulfide bonds turn out to be absolutely critical for the overall function of insulin because in the next steps what happens is there's proteolytic cleavage that cuts in a couple of places. It actually cuts segments out. So you can see this entire segment from here to here is removed by proteolytic cleavage, and we end up with an insulin that looks like this. Without those disulfide bonds, those two polypeptide chains don't stay in close proximity like that, and as a consequence, would have no function. So insulin can be processed in this way, and it gives us a molecule that has two polypeptide chains, even though they both were on the original same polypeptide chain. There are many post-translational modifications that happen to proteins. Okay? Some of them involve proteolytic cleavage. Some of them involve putting phosphates on, as we talked about. Some involve taking phosphates off. You saw how the collagen, for example, got the hydroxyproline, the hydroxylysines. Those are post-translational modifications. So a lot of examples of post-translational modifications that occur inside of cells. We talked also a little bit about the stability of messenger RNAs. If you recall, I said when we looked in the bacterial cells that the typical half-life of bacterial messenger RNA was a few minutes. whereas in a human cell, typical messenger RNA might be around for 24 hours. So we saw some very big differences in how long those were there. And the reason that we saw those differences really had to do with different structures of the messenger RNAs in prokaryotes versus eukaryotes. And the fact that, but the important fact in both was that there are nucleases, known as RNases, that are chewing those messenger RNAs up. Okay? Cells don't want to keep anything except DNA around for the long term. Okay? Maybe the membranes. Okay? The membranes, I guess, are important. But other than the membranes and DNA, the cells don't want to keep anything around for the long term. And the reason is that we could imagine that damage occurs over time. We might have, let's say, if we have a messenger RNA that has incorrectly copied a DNA, we don't want that hanging around for a long time and making bad protein as such. So if it's only around for a short period of time, then the effect is smaller. All right? So in addition to stability of messenger RNAs that we see, there's also stability issues for proteins. And proteins can, in fact, get chemically altered without, not post-translational modifications, but they can be damaged. Okay, so we think about, for example, reactive oxygen species that we've talked about. Reactive oxygen species can damage amino acid side chains in proteins. And if they damage amino acid side chains in proteins, we might have a protein going haywire. Okay? Some proteins going haywire are really bad. Okay? They can induce a cell to divide uncontrollably, causing a cancer. So the cell, to ensure that it doesn't have some rogue protein or rogue messenger RNA around for a long period of time, will typically degrade them on a fairly regular basis. What you see on the screen is one scheme that cells use to degrade proteins. Okay? And it seems like, wow, that's not very efficient. They make this protein, then they turn around and degrade it. And yeah, that's true. But again, it's for the reason that we don't want to have these problem proteins floating around, that we have to basically recycle everything that we make. Well, the way this is handled is by the attachment of a little flag that goes onto a protein. That little flag is a small peptide called ubiquitin. Ubiquitin is a tiny peptide that gets put onto a protein. And when it's on there, it's literally a flag, because that ubiquitination, as it's called, results in the protein being targeted for degradation inside of a complex called a proteasome. Proteasome. Okay? So proteasome is designed to basically take things that have been targeted for degradation and degrading them. Well, if we think about it, what's happening in that degradation inside of a proteasome, what's happening is proteases are chewing up that protein. And the product of chewing up protein, of course, is amino acids. The amino acids can then be either metabolized further, or they can be used to make new proteins. So there's a recycling that's happening with these. And so proteins that you have in your body, the amino acids in one protein might have a few hours previously been part of another protein. And that's possible to have happen because of this recycling process. So the proteasome plays a very important role in that recycling of proteins.
Did you say ubiquitin is specific for individual proteins, or is it just...
No, it's a general small peptide. It's not specific for any individual proteins, no. ubiquitin. It's right here on the screen, and it is right there where my thing is. Everybody's squinting. ubiquitin. Ubiquitin. And the reason it got its name was when people first discovered it, they found it everywhere. And somebody said it's ubiquitous. And so the name stuck, and that's why it got called ubiquitin. And there's a lot of ubiquitin inside of cells. Other considerations about proteins, and this is a really interesting one. Okay? I mentioned it earlier in the term, and it's called the 21st amino acid. The 21st amino acid. And this guy is a legitimate amino acid. It's called selenocysteine. And we have a few proteins in our body that use this amino acid. Okay? We use this amino acid. Well, how does it get in? The other ways we've seen, besides the 20 amino acids, we've seen modifications, for example. So we have hydroxyproline in our collagen because we covalently modify that and put it in. Well, we don't covalently modify this guy and put it in. We actually put it in as selenocysteine. So, well, the genetic code only specifies 20 amino acids. How do you get a 21st amino acid in there? And it's kind of an odd trick that happens. Okay? One of the stop codons, and sometimes the tryptophan codon, which is actually related to those, will get charged. Okay, I'm sorry, one of those stop codons will have a rare tRNA that will have this guy attached to it. Okay? And during the process of translation, at a very low frequency, this guy will get put in instead of translation stopping. Okay? Now, if you think about this, this is really odd. Okay? That if translation stopping occurs, that is, a stop codon is encountered and everything falls apart, then we never get this into our proteins. Okay? But at a low frequency, some proteins actually get this into them. And not only do they get it into them, they need it to function. Okay? They need it to function. Now, I won't go into all the reasons why this happens, but I will give you a sort of an example of something that you've actually seen that tells you a little bit about how that can happen. Think about how the cells make the termination of transcription occur when there's no factor. How do they do that? Transcription independent termination. How does that occur? So transcription termination factor independent occurs by the stem loop, right? We saw the base pairing occurred within that RNA. And so you've seen that RNAs can... Did you lose something?
It's okay.
Okay. Hope I don't step on... Oh, there it is. Hopefully, yeah. So we have pairing that can occur within a messenger RNA. It turns out that these guys that incorporate these selenocysteines near that stop codon have an unusual structure that forms, and it confuses the ribosome. And the ribosome doesn't see that stop codon as efficiently as if that structure isn't there. And so at a low frequency, they actually get this into them as a result of that. And as I said, the proteins that get this actually need this as part of their catalytic action. So selenocysteine is a very rare, it's very uncommon amino acid, but it's one that we have in our body, it's one that we need. And it's actually why, from a nutritional perspective, why we need selenium in our diet. Okay? We need selenium in our diet. Selenium is a really toxic compound. It's a really toxic compound. So it's one of those micronutrients you don't want to take too much of. And I, myself, would probably never take selenium on a routine basis just because of the toxicity issues associated with it. If I knew, for example, there was some issue that could be fixed with selenium, then I would consider that. But in general, selenium is pretty hard to get a deficiency in the diets that we eat. Okay, chaperones, I've talked about those briefly. Chaperones, you may recall, are these complexes that help a polypeptide chain to fold properly. And I've talked about those previously, so I'm not going to go into that here. You see, in E. coli, this is a chaperone system. It's called Gro-EL, and it has a little lid called GroES. And what the chaperone does is it actually allows the polypeptide chain to get inside this chamber and not interact with anything else. And so it gets a chance to fold on its own without being interfered with by other proteins or other molecules. Okay, so much for translation. Why don't we finish it up with a song, and then we'll move on to biotechnology. So this is a much easier song to sing today. It's to the tune of, it's not to the tune of Good Protein Synthesis, it's to the tune of Good King Wenceslas. I just noticed that. Good King Wenceslas, okay. Da, 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 right? Okay. Amino acids cannot join by themselves together. They require ribosomes to create the tether. All the protein chains get made according to instruction, carried by mRNA and peptide bond construction. Small subunit starts it all with initiation, pairing up to RNAs at the docking station. Schein-Delgarno's complement in the 16 S's lines the AUG up so synthesis commences. Elongation happens in ribosomic insides where our RNA creates bonds for polypeptides. These depart the ribosome, passing right straight through it in the tiny channels there of the large subunit. Finally, when the sequence of one of the start codons parks itself in the A site, synthesis can't go on. P site RNA lets go of what it was holding, so the polypeptide can get on with its folding. Okay, a little easier to sing. Da, 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 da. Okay, so biotechnology techniques. Okay, biotechnology arises from some of the really clever techniques that are here. And I may have told the story in class. If not, I'll repeat. I'll say it here. If not, if I have said it, I will repeat myself. But I'd like to give the example of the way in which biotechnology has revolutionized our world, literally in my lifetime. So back in the dark ages of the early 1980s, I was a graduate student here at Oregon State. And the biotechnology world that we had then was very, very different than the one today. And I was working on a project in a lab, and I was getting to the point in about the mid-'80s where I had to settle on what I was going to do for my final PhD thesis. And I talked to my mentor, and he wanted me to isolate the gene for a human protein. It was actually for a topoisomerase. That's what he wanted me to work on. And one of the fortunate things is that you get to sort of negotiate these sorts of things with your mentor. And I gave it some consideration, and I finally told him, I said, you know, I just don't think I can get this done in the next couple of years. It's just too technical of a problem. And at that time, isolating a human gene was a pretty advanced PhD project. Okay? Today, I could take any of you, and tonight you would have that same gene isolated. Okay? You wouldn't have to have any training. You wouldn't have to have anything else. I could take you and show you how to do that. You could have it, and in the morning you'd have the gene. Okay? Now that tells you something about how simple the techniques of biotechnology have become. And so when we think about when I was a graduate student, we didn't think it was possible. We literally didn't think it was possible to sequence the human genome. People were starting to talk about that, and we said, that will never happen. Well, these techniques, never say never, of course, these techniques got an amazing evolution that they went through, and as a consequence of that, really changed molecular biology immensely. So the power of these techniques I'm going to show you is really hard to overstate. I mean, they're pretty incredible. Well, we start with some fairly basic things, and some of the things that we take for granted. And I'm going to start with restriction endonucleases. Okay? So restriction endonucleases are enzymes. They're also called restriction enzymes. Okay? And they are actually bacterial enzymes. We don't have them in our cells, but bacteria have them in their cells. And these enzymes have a really, really, really useful property. This is one of the first things that made modern biotechnology possible. When I was in the lab, these were just starting to become available. Okay? What do? Well, they recognize a specific sequence on a DNA, and they cut it. They recognize a specific sequence, and they cut at that sequence. Well, what that means is that you can cut DNA into discrete pieces wherever one of those sequences appears. Okay? The sequences tend to be about anywhere from 4 to 8 nucleotides long. Most commonly, they're about 6 nucleotides long. And they give us the ability to manipulate DNA in a very, very defined and easy fashion. Well, why do bacteria have these enzymes in the first place? Okay? The bacteria have the enzymes as a very primitive immune system. Okay? So I'll tell you briefly about them. I won the prize. A very primitive immune system, okay? Well, in our body, we have antibodies. The antibodies recognize invaders. They attack it. They kill it. They, dispose of it and all that sort of stuff, right? Well, bacteria are single cells, right? We have a bloodstream that carries all this stuff around. Bacteria are free agents. Every bacterium for themselves, right? Well, bacteria get infected with viruses just like we get infected with viruses. And in the case of bacteria, they get infected by things called bacteriophages. Or more commonly, you're probably going to call them phages, P-H-A-G-E-S. Phages are bacterial viruses. Well, if I'm a bacterium and a phage comes along and attacks me, it would sure be nice to try to fight the thing off. Well, I don't have an immune system, because an immune system takes multiple cells, it takes a bloodstream, it takes all this sort of thing. So I've got two possibilities. One, I fight it off, or two, I'm totally hosed. Well, they're frequently hosed, but bacteria have evolved a system of protection that involves restriction enzymes. And so I'm going to tell you about that. All right? Restriction enzymes, you can imagine if this virus in its infectious cycle injects DNA into the bacterial cell, what's going to happen is if you have restriction enzymes in there, if those sequences that the restriction enzyme recognizes and cuts are in the viral DNA, you've just cut up the viral DNA and presumably protected the cell from this invading virus. That's pretty cool. Until you think, well, how is it that it doesn't cut up its own DNA? These are fairly short sequences, and we would expect in a genome of 6 million base pairs that on a random basis, these sequences would appear several thousand times. Okay? So how is it that they don't cut themselves up, to cut up their own DNA? Because that would not be, that'd be, I'd say throwing out the baby with the bathwater, but it would be more like killing the baby to save the baby, right? I mean, you're not going to do anything with this. Well, it turns out restriction enzymes are half of a system called restriction modification system. So the restriction system is what you've just seen, the cutting up of the DNA. The modification system is what protects. So the modification involves a second enzyme. So in the bacterial cell, this enzyme works, and it's called a methylase, meaning it puts methyl groups on things. M-E-T-H-Y-L-A-S-E. A methylase puts methyl groups on the exact same sequence that the enzyme wants to cut. Well, okay, you probably can start to see the strategy here. If the sequence has a methyl group on it, the enzyme won't cut it. The enzyme won't cut it. So the cell protects its own DNA by the methylase running around, and every time it hits one of these sequences, it puts a methyl group on it, and the restriction enzyme, for all it wants, can't cut up its own DNA. Is it a perfect system? No, it's not. There is no system that's perfect, and this system is not perfect either. Okay? How could you imagine this system might not be perfect? Yeah?
Well, when I was reading about the repairs to DNA,
Yep.
it said that there was the new sequences, the new replicated ones, weren't methylated yet, and that's how it recognized the change.
Okay, so that's actually a different. You're talking about a different process, but you're exactly right. So what she's talking about is a different kind of a different methylation that's done for the cell to be able to tell which strand is the newly synthesized one. And you're exactly right. That's an identification thing, and this is a protection thing. Okay, yeah.
Well, I don't understand the benefit of it, but in effect, it's protecting from splicing it. Is that what you're saying?
It's protecting the virus from coming in and being able to do its thing, because it's chopping the virus DNA into pieces.
And preserving itself.
And it's not chopping up itself into pieces. So what might be some holes in this system?
It's just a methyl group on the invading virus.
Yeah. So what if the methylase gets to the virus first? Now the virus is protected, right? Okay. So we can imagine this probably wouldn't be a good career move for the cell. And so, yes, viruses do sometimes escape it by having the methylase, by encountering the methylase before they encounter the restriction enzyme, and we could imagine that that's a factor. So yes, that's one of the ways in which that can occur. Okay? That's kind of a cool system. So what we do in the laboratory is or at least companies do this, we don't do it anymore, we used to purify them ourselves, But companies have purified the restriction enzyme and throw the methylase away because you don't want to have the methylase. You want to cut up a bunch of DNA, right? So you take the restriction enzyme and you cut it. So here's how it works. Here's one enzyme that recognizes a sequence GAATTC. And if you look at this on the bottom strand, it reads the same way, GAATTC. Most restriction enzyme recognition sites are like this. It's what people call palindromes. It's not technically a palindrome, but for a molecular biologist, that's a palindrome, reading the same way on the top strand as it reads on the bottom strand. Well, the beauty of this is that the enzyme, all it has to do is recognize GAATTC. It means it cuts it here. It also cuts it over here. In this case, it's cutting and it leaves what's called an overhang. This overhang is here, and the overhang is there, right? Because it cut between the G and the A, and it cut between the G and the A. Well, that's kind of nice. If I take a DNA and I cut it up with EcoR1, it's always going to, this is an enzyme called EcoR1. If I cut this guy up, I'm always going to have overhangs that look like that. Well, the beauty is, if I decide I want to put the pieces back together, they go back together really nice because look, here's a new fragment that has that same overhang, AATT, and look, AATT will base pair, and the DNA ligase says, oh, look, everybody's happy, I join it together, and I'm done. Well, what I've just done with DNA ligase is I've taken a new fragment and I've joined it to an old fragment, and I've made a recombinant DNA. So when I match up my sequences like that, they'll usually have, these are called sticky ends, is what they're called. Sticky ends are overhangs that can form base pairs, as you just saw right here. Okay? When that happens, I have a nice way of putting the pieces back together and making a desired molecule. DNA ligase, I'm not enunciating that very well. DNA ligase is very, very important in the making of recombinant molecules. Very, very important. Now, okay. Now, what if the restriction endonuclease cuts right here between the A and the T and the A and the T? What's going to happen? Do I have a sticky end? I have no overhang, right? So some enzymes cut right in the middle of the sequence like that, and they create what's called blunt ends. There's no overhang. And the reason we know that overhangs are really useful is if we try to ligate together blunt ends, it's much less efficient. They don't go together very well. Okay? It's much easier to ligate sticky ends than it is blunt ends. We can still link them together, but the efficiency goes way, way down. So those sticky ends really do make a difference in helping us to join pieces together. Okay, what you've just seen, you've seen a restriction enzyme, you've seen a ligase, and now you've made a recombinant DNA molecule. Okay? Bang, bang. That's simple. It was the first thing that attracted me to molecular biology was that thing right there, being able to make that molecule. Okay? This is showing you what I told you before. There's the unmodified DNA. Methylase hits it. There's the methyl group sitting on the bases there. And this guy will not be touched. Bacterial. Oh, boy, look how she spelled that. Endogenous bacterial DNA. It should be endogenous bacterial DNA. Okay, I get amusing things out of this. You guys are the guinea pigs for this. You know, I'm sorry. Should have those fixed. All right. And yes, she speaks English as her native language, so I'm not sure what she didn't proof that very well. All right. So we see the protection that happens as a result of the methylase coming in there. Okay. Now, there are hundreds of restriction enzymes that are known. And they have unique sequences that they recognize. Okay? Well, we're not going to memorize the sequences. That's kind of a waste of time. But you get the idea that, you know, if I try to cut up one DNA and it doesn't cut, perhaps that sequence I'm trying to, or the enzyme I'm trying to cut with, that sequence doesn't occur, I can try a different DNA and, you know, do that. So I have all kinds of options for different enzymes I can use to cut up DNA. Well, that gives me a lot of latitude in doing things. I talked about making a recombinant molecule, and I sort of diagrammed that out for you just a second ago, but now I want to spend a few minutes actually making a useful recombinant molecule. So you've seen, for example, that having some fragments with sticky ends really useful. And having DNA ligase is really useful. But let's say I want to make a recombinant molecule, but I want to be able to do something with it. I want to be able to use this for something. Let's take an actual example. Okay? Let's imagine that I want to get a bacterium to make human growth hormone. Okay? Human growth hormone is pretty difficult to isolate from human blood. It's extraordinarily expensive. It takes thousands of gallons of blood to get enough to actually treat somebody with. What if I had the gene for the human growth hormone, and I wanted to get bacteria to make that gene? What would I have to do? Right? Well, I would have to have on a restriction fragment that gene of interest. Right? So let's say I cut with EcoR1, and I've got my gene of interest on EcoR1. Okay? Well, if I just take that fragment and I put it into bacteria, nothing's going to happen. Because, A, when the bacteria divide, the DNA is not going to divide because it doesn't have a replication origin. It's going to basically disappear. Right? So what I would like to be able to do would be able to have that gene be in something that will replicate in bacteria. I would also like for that gene to be able to be transcribed and translated in bacteria. And third, I would like to be able to select for the bacteria that get this gene. Okay? Because not all bacteria are going to get things that I put into them. In fact, when I try to put things into bacteria, at the very highest rate, I get about 1 in per every 100,000 cells. Very low efficiency of getting DNAs into bacteria. So I've got to think about these things if I want to get a bacterium to make my desired protein. All right? Well, to do this, we use something called plasmids. Okay? Plasmids are circular DNAs, and they're circular DNAs that have a replication origin in bacteria, meaning that they will be replicated. Okay? So the first useful feature is they have a replication origin, and when the bacterium replicates, these guys will replicate. Okay? Well, that's fine and dandy. Let's imagine that I take my plasmid. Forget about the gene for the moment. I've got a plasmid, and I'm saying I've got this plasmid. I want to put it in bacteria, but only 1 in 100,000 bacteria are going to get this plasmid. How am I going to fish that one bacterium out that has the plasmid from the 99,999 who don't have that? Okay? Well, this is where we use a process called selection. It means that the plasmid that I put in isn't just a bare plasmid that has nothing in it except an origin. I put in something that has what's called a selectable marker. A selectable marker. Gesundheit. What is a selectable marker? Well, a very simple selectable marker that we use in the laboratory is a gene for resistance to an antibiotic. A gene for resistance to an antibiotic. Meaning that if a bacterium gets my plasmid, it gets that gene, and now it will be resistant to that antibiotic. And I can identify the bacterium that gets it by the fact that it will grow in the presence of that antibiotic, whereas those that don't get that plasmid will not. They will die. So anything that's living, when I give it that plasmid, is clearly something that's gotten the plasmid in it. So 99,999 of them are going to die. 1 is going to live, and it's going to thrive. It's going to keep growing because the antibiotic is not going to stop it from growing. Okay? So I've got a selectable marker. I've used that selection to help me identify the bacterium that gets it. Well, that's fine and dandy. Let's imagine now, okay, I don't want to just put the plasmid in there. I want to put the plasmid in there with my gene. That's what's being shown here. So here's the plasmid. It's got an origin. It's got a selectable marker. And now it's got a place where I can cut it and put in sticky fragment of my gene. Okay? Do you suppose I want to put this in just a random place in this plasmid? And the answer, if Kevin says, do you suppose you want to, probably he means no. All right? There must be something more to it, and that's right. What would be the considerations of where we would want to put this in this plasmid? what would be useful characteristics for this plasmid to have? Based on what we've been talking about the past week or so. Yeah. So the origin replication is there, but that's not going to affect my gene. It'll affect the plasmid definitely. Yeah. Near a promoter. Beautiful. I put it near a promoter, then what's going to happen? It means that messenger RNA is going to be synthesized, right? Okay. What else might be useful for that to be right next to? Maybe a Shine-Dalgarno sequence? What's the use of the Shine-Dalgarno sequence? We didn't have to study for that for the test. I don't remember, right? The Shine-Dalgarno sequence is a place in bacteria where they know they line up where the AUG is. So now they'll have this RNA that's got the Shine-Dalgarno sequence on it, and it's going to tell it, start translating right here. So when the ribosome gets it, it's going to translate that messenger RNA, and since the genetic code is the same in bacteria and it is in humans, the bacterium is going to start cranking out human growth hormone in prodigious quantities. Dirt cheap. You feed bacteria nothing. Yes, sir?
So you'd have to isolate the introns or the exons?
Very good question. So he says, do we have to consider the introns and exons for this? And the answer is yes, we do. Okay? So ideally, we've taken something that's already had the introns and exons, or the introns removed, and only the exons are left. Yeah, very good point. Very good point. Yeah?
I thought you said that didn't happen in prokaryotes.
That's right. But if I put in something that has introns and they don't get removed in prokaryotes, then when they go to translate, those introns are still going to be there.
DNA.
Oh, so this is producing before eukaryote then?
I'm sorry?
So, because the exons are the actual ones we want.
The exons are what you want. And if I leave the introns in there, when the prokaryotes get it, they're going to be translating those introns, and that's going to screw up my protein. You see what I'm saying?
Yeah.
Good thoughts, yeah.
Would there be multiple Shine-Dalgarno sequences, considering that you also have to turn on the antibiotic resistance as well?
Good question. So will there be multiple Shine-Dalgarno sequences in this plasmid? And you're exactly right. So the antibiotic resistance gene already has that set up in it. So that's, but yes, it would have to have that in order to translate that protein that's going to give the antibiotic resistance. Exactly right. Yep. Good. You guys are thinking about this. This is great. Okay. Everybody understand that? So I just described to you. Okay. This guy, we call it a plasmid, but it's more than just a plasmid. A plasmid is just a circular DNA that replicates in cells. This guy has a better name. It's called an expression vector. So the expression vector will have the following things in it. It'll have an origin. It'll have a selectable marker. It'll have a promoter. And it'll have a Shine-Dalgarno sequence. Specific for the promoter. It'll also have the one for the antibiotic resistance gene. Okay? So now, when I put this guy in the bacterium, this is really simple. I just treat the bacterium with the antibiotic. I get the ones that I want, and then I just start collecting my protein. All right? One of the reasons that the biotechnology industry is growing with leaps and bounds is it doesn't take much bacteria to make a lot of really useful human protein. Okay? Pretty cool. All right. Let's talk about the word cloning. We hear the word cloning all the time, and I use it myself, and what a lot of people don't realize is the word is used in many circumstances. Many. When I was working in that PhD, my boss came to me, and he wanted me to get this gene from human cells, and his comment to me was, we should clone that gene. What does it mean to clone a gene? Right? We talk about cloning a sheep. We talk about cloning a human. We talk about cloning things. Isn't cloning, something that's really something pretty elaborate? A clone simply means identical copies of. That's all it means. So if I'm cloning a gene, I'm isolating multiple copies identical of that gene. No? Oh. I thought you were saying no. Yeah, it is. I was going to argue with you. Punch it. Okay. So that's all a clone means. So a clone, if I had, for example, how many people have streaked for bacteria, right? You streak for bacteria, you take microbiology, right? You're making clones. Because when you streak that thing out, you're smearing it out so that you get an individual cell, and that individual cell grows, and when it duplicates, it makes thousands and thousands of copies of itself, and you've got a clone. Every colony, therefore, is a clone. If I take one of the nuclei of my cells and I put it into an egg cell, all right, then that egg cell grows up. All those copies will have the same DNA as me. They'll be a clone of me. Okay? So clone simply means that, just many, many copies of something. And people use it as a verb. I'm going to clone this. All right? I'm going to clone this means I'm going to isolate many, many copies identical of something. Okay.
That's the improper use of grammar.
It is pretty much an improper use of grammar, but you'll hear it used all the time, yes, including by me. All right, so let's see. I'm trying to think of something that'll be useful for you here. Oh, here's a good one. Okay. Oh, I got to fix that link for you. What's wrong with that? Oh, I know it. I'll fix that for you tonight. Okay, let me tell you what it is. That's kind of cool. All right. So I talk about having a screen, a selectable marker, right? Well, a very easy selectable marker is an antibiotic resistance gene, because I can treat with antibiotic and I get what I'm after, right? Well, just like it's very inefficient for me to put DNA into bacteria, so too is it inefficient for me to put that new fragment into that cut plasmid. Okay, so I'll go back to the slide and show you. All right. When I'm trying to make this recombinant guy right here, okay, well, it looks like it's fine and dandy. I cut, I've got this, I ligate, I get this, but maybe 1 in 1,000, maybe 1 in 10,000 gets that insert. The most common thing that will happen is this will just ligate back with itself. Nothing will go in there. Okay? So I sure would like to have a way of knowing if I get my insert in there or not. Okay? So this gives us another consideration for selection. Okay? And this uses a gene that you've already seen already. Remember the lac operon? The lac operon, I told you, had genes that allowed a bacterial cell to metabolize lactose. Okay? Specifically, they have a gene called beta-galactosidase. And yes, you should know this name. Beta-galactosidase. G-A-L-A-C-T-O-S-I-D-A-S-E. Beta-galactosidase is a name of an enzyme. And what it does for bacteria is it takes the sugar lactose and it cuts it in half. We'll talk about it later, but it cuts it in half. All right? Well, that doesn't really give us anything to select because neither lactose nor the products of the cutting in half tell us anything. We can't see them. So scientists have devised artificial substrates that look like lactose. And they fool the enzyme. Remember the biochemist being lazy? We had the one earlier that made the yellow color. Okay? Well, biochemists have created a compound called XGAL, the letter X-GAL. And it fools the enzyme. And when the enzyme cuts it, you get a blue color. Okay? So let's go back to that plasmid. I got a plasmid that's got an antibiotic resistance gene. It's also got beta-galactosidase gene in it. If this plasmid gets into a bacterial cell, it will be resistant to antibiotic. And if I give this cell XGAL, it's going to turn blue. Because any cells that have the plasmid are going to make the beta-galactosidase, and the beta-galactosidase is going to cut XGAL and make blue color. Well, that's cool, but I don't need two things to tell me it gets into cells. All right? But here's a cool trick. The XGAL is set up so it's got restriction. I'm sorry, the beta-galactosidase gene is set up so it has restriction sites in it that I can cut. One of them is EcoR1. Now, let's imagine here is the beta-galactosidase gene, and right in the middle of it is an EcoR1 site. And I take and I cut with EcoR1 and I insert my fragment. If this guy comes back together, I'm still going to have a functional beta-galactosidase gene. I'm still going to have blue color, right? So all the ones that don't have a fragment are going to have a blue color. But the ones that have a fragment will no longer make a functional beta-galactosidase gene. They won't have a blue color. They will have a white color. Because there's no beta-galactosidase to break down the X-gal that I give them. And the figure I will get will show you this. I'm telling you just now so you'll know what you're looking at when you get the figure. Okay? When I give these cells that have this guy, X-gal, the beta-galactosidase gene got interrupted. It's not functional anymore. It's lost its function. So only those that have an insert will make colonies that are white when I give them X-gal. It's called blue-white screening. And it tells me not only did the plasmid get into the cell because it's growing in the presence of antibiotic, but that plasmid has an insert in its beta-galactosidase gene, and since this is the thing that I was inserting, bingo, I got what I want. Make sense? Yeah, clear as mud, right?
So if it doesn't turn blue, we've got the proper sequence.
If it doesn't turn blue, I've got just what I'm after. You got it. Pretty cool. Yeah.
Have you seen the glowing animals?
Have I seen the glowing animals? I have. I have. I've even seen, this was a very cool thing, they made glowing Christmas trees. So you could spray the stuff on Christmas trees, and they have a gene called luciferase that gives light. So you spray the tree with luciferase, and the tree lights itself. It's kind of cool. Yes, Chris? How do you separate the blue colonies from the white ones? I'm sorry? Well, actually, no. Remember that we're streaking these out, so they're growing up as single colonies. So a white will be fully white because once it starts white, everything that's duplicated from it will still be white. So if it's smeared with other ones, then I have to streak it out a little bit further to get it. But streaking is the way that I would get that. Make sense? Okay, so that's it for today. Thank you, and I will see you on Friday. Yes, sir.
Ubiquitin, a peptide.
Yep.
Isn't a peptide an amino acid?
A peptide is a sequence of amino acids joined together.
Oh, okay. It's a
Right?
sequence of amino acids.
Right.
Okay.
It's like a protein.
So it's just
tiny.
a protein flat.
Yeah. Yep.
So that's when it comes to when you're talking about inserting the plasmid.
Yep.
So it can insert with sticky or blunt end. It's just the sticky end.
It can. Stickies are much more efficient.
So if you have, how do you know if you have a blunt or stickies based on what you cut it
Okay,
with? Or-
that's right. So the enzyme will always give the same characteristic end. So if you cut with EcoR1, you'll always get a sticky end.
Okay, cool. Every
If you
time.
cut with something called small one, it'll always give you a blunt end.
So you know when you're doing that, you're always cutting with you're always going to get a
That's
sticky
right.
end.
You know what you got.
Awesome. Thank
Yep,
you.
you bet.
So with selenocysteine, proteins that need it for a good function,